Dave says, yeah, I work with Robin, but this doing neuroimaging of psychedelics, but this is nothing to do with the neuroimaging. In fact, I'm not really going to be talking about global brain effects of psychedelics much at all. I'm going to be talking about molecular level, receptor level interactions, and what happens to the brain, what psychedelics do in the brain receptor level. And my talk's called Affinity and Beyond, purely because people tend to refer to the affinity of psychedelics for receptors as being the kind of main indication of potency. And while it's very important, there are other factors involved too. I'm just talking about classic psychedelics here, not about NMDA antagonists or anything else. So your sort of serotonin active classic psychedelics. So, as you know, psychedelics fall into two main categories, phenylethylamines and tryptamines. Here are some phenylethylamines, 2CB, DOB, 2CE, DOET, and I put the dosages up here. These dosages are just taken from PCAL and TCAL from Shulgin's books as just a standard reference. Now what you should really know here is that the difference between 2CB and DOB and 2CE and DOET is that there's an extra carbon on the alpha methyl here, which turns the structure from being a plain phenylethylamine into amphetamine backbone. That's the only difference. And you can see that difference creates a difference in potency of a factor of about 10. So DOB is 10 times as potent as 2CB. That holds true for the other halogenated phenylethylamines. So DOI, 10 times as potent as 2CI, DOC, 10 times as potent as a 2CC. Slightly different with 2CE. You can see that the potency difference is only about 5 or 6 times greater. But maybe I'll go into that a bit later. I'm not sure I'm going to have time. The other thing to say is that there are quite big qualitative differences. Although the dosages between 2CB and 2CE look very much the same, if you read the sort of online anecdotal reports, people tend to regard 2CC being much more powerful and a much more heavyweight psychedelic experience, closer to LSD. Here's some tryptamines. I'm sure you all know these. Psilocin, one of the active ingredients of magic mushrooms, 5-methoxy-DMT, another naturally occurring hallucinogen, and LSD. Again, the dosages, and again, big qualitative differences between psilocybin and 5-methoxy-DMT. A few milligrams of 5-methoxy-DMT by IV or smoked tends to be a lot more powerful than a few milligrams of psilocin by IV. And then finally LSD, with its, again, a factor of increase of potency of at least 10, so it's about 20 times more potent than DOB or DOI. And I guess the kind of the question here is, why are there these differences? What makes one chemical much more potent than another, and why are these qualitative differences? I'm going to try and explain what we think's going on. It's all very cutting edge, and we don't really know what's going on, but I'm going to try and explain it. So what are those receptor targets of psychedelic drugs? As several people said during this conference, 5-HT2A is the main receptor target. That's what we believe responsible for most psychedelic action. And there are plenty of studies, animal studies with rats and mice, showing that if you block activation at 5-HT2A, you can block the psychedelic response with LSD or DOI. And there's tons of work showing this. There's also a couple of human studies. Franz Vollenweider and his group did giving a ketanserin 5-HT2A blocker to humans and blocked the effects of psilocybin. So there's very strong evidence that 5-HT2A is the main psychedelic receptor. 5-HT2C, it's got a very similar structure to 5-HT2A. I think 80% homology in the genome. I'm not quite sure, but it's very, very similar in structure. So most psychedelics have activity at both receptors. Generally, the 2C activity is about 2 or 3 times weaker. And while 2C isn't regarded as being able to cause psychedelic effects on its own, it probably modulates the experience somewhat. And finally, 5-HT1A, tryptamines, but not phenylethylamines, have activity there. The action can actually reduce psychedelic effect. Rick Strassman gave a 5-HT1A blocker and found that it increased the potency of DMT given to human subjects. And it's also, actions are also anxiolytic. There are lots of other receptors involved, depending on the drug. LSD, for instance, has lots of effects on dopamine receptors, adrenergic receptors for the phenylethylamines as well. But they're generally regarded as just adding sort of colour to the experience, but not really producing the main psychedelic effect. Right, now I'm going to talk about affinity and efficacy and the differences between the two and the relationship between the two. Affinity is a drug's capacity to bind to a receptor and measure its stickiness. Robin said this in his talk yesterday, and Dave Nichols once said to me that it was like throwing a ball at a wall, and if the ball bounces off the wall, then it has low affinity. But if it's like a clump of mud and it sticks to the wall and falls down slowly, it has a higher affinity. It's a pretty crude analogy, but I think it helps to understand. It's normally expressed as a Ki value, which is the dissociation constant. And that's the concentration of the drug where half of the receptors you're interested in are occupied. So lower Ki value equals better affinity. Oh, I should say that what's actually going on is quite a complex equilibrium state. Receptors, drug molecules and receptors are binding and dissociating the whole time. So when you're looking at a stable equilibrium, that's kind of the average of what's going on. Efficacy, a drug's capacity to produce a biological response. Now, that biological response can be a number of different things. When we're looking at neurons, it might be an increase in the neuron firing, it might be changes in gene transcription, it might be secondary messengers and other chemicals inside the cell. That's expressed as an EC50 value, the concentration of the drug that produces half the maximum response, and also an intrinsic activity. There's the maximum percentage the drug produces compared to the natural agonist. In this case, the natural agonist is serotonin. So serotonin would produce 100% response, and other drugs would produce on a sliding scale. So a full agonist was around 90 to 100%, a strong partial agonist sort of 50 to 90, a weak partial agonist less than 50. It used to be thought, and any pharmacologists in the crowd who want to correct me on this, it used to be generally thought that receptors could take one of two states, active or inactive, and an agonist would shift the balance to being more active receptors, whereas an antagonist would bind to receptors but not shift the balance in any way, so it wouldn't increase the response, whereas an agonist would. But that's now slightly changing, and part of what I'm going to talk about today is how receptors can be in multiple different states, not just active or inactive, but activating different pathways in different ways. These are some experiments now. This is some data from three different, two papers and one PhD thesis. This is Glennon, 1984. It's a very famous paper. Robin showed this chart in his talk yesterday. It's influential in helping show that 5-HT2A was the primary receptor for psychedelics. Tom Ray's paper, 2010, quite controversial paper, and then Michael Braden from Dave Nichols' lab. This was from his PhD thesis, but it's been published in several papers as well. So this is Glennon's results. So you can see on the x-axis is the dose. It's a log 1 over dose. So the further up the scale, you get the more potent the drug is, and affinity the same. The further up the scale, the higher the affinity for the drug. And you can see that's a really great correlation, R of 9.3, R squared of 9.3, but there are several things to sort of try and understand this graph and try and understand what the results mean. Firstly, this is done with rat receptors. It's whole rat brain homogenate was used in this experiment, so it's not human. And rats and humans, although they have very, very similar 5-HT2A receptors, they're not quite exactly the same, and there are differences in affinity. The second thing to note is that this was done before the discovery of the difference between 5-HT2A and 2C. So actually this is a mixture of 2A and 2C, because they just didn't know that there were two different kinds of 5-HT2 receptors at the time. And finally, yes, any affinity experiment like this, you're always looking at it's a challenge experiment. You're always looking to see one drug has to be displaced by another. And in this case, the drug that he's using is ketanserin, which is an antagonist. So these results are showing how good the test drugs are at displacing ketanserin. And ketanserin as an antagonist binds to both active and inactive receptors. So really, to get a better idea of what makes a drug more potent, you should be looking at just how it binds to active receptors. So although it's a really good correlation, there's nothing wrong with the experiments. It can be slightly misleading just to say better affinity, better potency. And also the other thing you might notice is that all these drugs, with the exception of LSD, are all of the DO class. They're all amphetamines of different kinds. So in one sense, it's not that surprising if they fall on such a nice, neat line, because they're all of one particular group, and LSD's out there on its own. So enough about that. Quickly going to show you Thomas Ray's experiment. This is a much better spread of drugs. You can see we've got 5-methoxy-DMT, we've got psilocin in there, we've got a bunch of the 2C drugs as well, LSD, analog, TMA2. So this is a better spread of drugs, but This was regarded as being quite controversial at the time because a lot of the data was flying in the face of stuff that had been previously published. And some people thought it was radical and was going to change our understanding of how the binding to receptors. But it turned out, as far as I know, that one of the authors took his name off the paper because he didn't believe that the experiments were conducted in a sufficiently methodical manner. So this is generally regarded as being a bit controversial. It's against ketanserin, again. I think it's rat, I'm not sure, but it's definitely versus ketanserin. So I'm just showing you that kind of to show that almost every paper you look at, there will be different affinities quoted, and they tend to vary quite a bit, so you have to kind of like try and work out who's done the best science and their methods and what they're doing to really kind of get a handle on this. So finally, here's Braden's thing, and I actually think this is probably the best out of the three. This is from Dave Nichols' lab. You can see that the correlation isn't as good, of 0.56, but still pretty high. That's a very high correlation for this kind of thing. And this is against DOI rather than ketanserin, so against an agonist. So this should give a better, this should tell you more about how drugs bind to the active state. And it's also human, cloned human 5-HT2A receptors rather than rat. And the things to know about this are, the interesting stuff here is that if you look here, the DOI, DOB, have almost exactly the same affinity as 2CI and 2CB, despite there being this tenfold difference in potency. And if you look down here, psilocin, you can see that its dose is about the same as 2CI and 2CB, despite having much, much lower affinity. So why is this? Clearly, you can't just say that higher affinity means higher potency. There's obviously something else going on. Before I talk about the efficacy of these receptors, I'm going to briefly explain to you about the two main different kinds of receptor on the synapse. And this is a simplification, but it's important to understand it, I think. So, ionotropic and metabotropic receptors. This is an ionotropic receptor. It's an ion channel. Agonists bind to the ion channel and open it, and it lets ions in or out of the cell, directly influencing whether the neuron fires or not. This is a metabotropic receptor, and this is the kind that 5-HT2A is. The drug binds to the receptor, and it sets off a cascade inside the cell, which starts with a G protein, and then moves on to a cascade of other messenger chemicals inside the cell. So this means that the response is slower and is also not necessarily localized so tightly in space. When I say slower, I only mean very slightly slower. Instead of it being milliseconds, it's a matter of maybe seconds. And it can be distributed further through the cell and either into other cells rather than just being immediately localized in that particular cell. This is a schematic of these cascades inside the 5-HT, inside the neuron. This is 5-HT2A receptor here. The drug binds at the top here. These three things are G proteins: alpha IO, alpha 1213, alpha Q. And they are involved in what you say is a very complicated signaling cascade. You don't need to worry about this too much. All you need to sort of know is that there are different secondary messengers that get produced, and the ones we're interested in here are this IP3, inositol triphosphate, and AA, which is arachidonic acid. Now, Nichols' lab did a bunch of experiments to investigate how psychedelics might change or might activate these different pathways. And this is some of their data. This is from another PhD thesis by Parrish, also from Nichols' lab. So they looked at these DO and 2C compounds. So you see DOB, 2CB, DOI, 2CI, DOTFM, and 2CTFM. And you can see here, this column here is affinities of the human 5-HT2A receptor against DOI. So you can see the affinities here are almost exactly the same. There's no statistical difference between the affinity of DOB and 2CB, or between DOI and 2CI. If you look at the EC50 values, this is for the IP pathway. So let's just go back. This is for the production of inositol triphosphate IP. So that's what this column is. So the EC50 values are also almost exactly the same, no statistical difference there. But the intrinsic activity is statistically different. So you see 70% for DOB, 40% for 2CB, 50% for DOI, 30% for TCI, and then 50-26 here. So that looks great. We've got a really good result there. It looks like having more IP production means more potent psychedelic. But unfortunately, it's not that simple. Here's another chart also from Parrish's thesis. It completely falls down when you look at tryptamines. You look at LSD, 5-MeO-DMT, psilocin. They've got these really high here. 5-MeO-DMT and psilocin have really high EC50 values, so their potency should be much, much less. But in fact, psilocin's potency is about the same as 2CB. So this is the other column we're interested in, the AA release here. So they theorised that actually what mattered was having greater AA release and favouring AA release over IP production. And that sort of holds true. You can see that like psilocin has better AA release, 86, so lower values are better here, and higher values are better here. And the same for DMT compared to its IP production. But it doesn't really hold true for LSD, for instance. Still pretty low IA, intrinsic activity there, 56%, despite this. So if you look at, say, DOB, you'd expect from this that DOB would be a much more potent psychedelic than LSD, when of course it's not. LSD is much more potent. So there's a sort of trend emerging here that psychedelics turn on these secondary messenger systems in the cell to a different degree than non-psychedelic agents, but the actual relationship between the two and which really matter, you can't really say from this data so far. So here's some more data from another group. This is Gonzalez-Meso, did 2 papers, 2007, 2009, where they're looking at LSD and lisuride. Lisuride is a non-psychedelic analogue of LSD. The only difference, it's got this extra nitrogen here on the tail, and it has pretty similar, if not quite identical, but very, very similar affinities and efficacies for IP production. So going on what we know so far, you would have thought that lisuride was going to be a psychedelic, but in fact it's not in man to any reasonable dose. So they gave, this is all in rats, yes, this is all in rats, I think, that were, so there's two sets of rats here. There's rats with normal 5-HT2A, and there's knockout rats who've got no normal 5-HT2A receptors at all. So the knockout rats have no psychedelic effect, basically, and these two things on the side are two different genes, CFOS and EGR2. It doesn't really matter too much about the details of these genes. I'll show you another diagram in a minute. But the important thing to see here is that the psychedelics up here induce both CFOS expression and also EGR2 expression, whereas the non-psychedelics, lisuride and ergotamine, only induce CFOS, they don't induce EGR2. So here's a revised image. This is from Gonzalez-Meso's papers, and this shows, again, a similar sort of scheme. Here's the 5-HT2A receptor, the drug binds here. These are the pathways, so the PLC one is the one that results in IP production, the SRC one is one ends up in AA production, and these are the two genes being transcribed inside the nucleus. So CFOS is the one that they both activate, and EGR2 is the psychedelic one only. And the great thing about this is it's full of question marks, as you can see. Like, we don't really know the details of all these pathways inside the cell at all, and there's a question mark over beta-arrestin, which I'm not going to talk about, but that's another secondary messenger that might be involved, and there's very probably several other secondary messengers yet to be discovered. We know about 4 or 5 in 5-HT2A. So how long have I got for time? I'm doing all right. Okay, so this is the kind of interesting stuff then. How do we get from these drug effects at a receptor level? How do we get from there to changes in neuronal firing on a local level? So how does that change the way the neurons fire, and how do they relate to each other? And then finally, how do we get to this, the changes in global brain activity that Robin was talking about yesterday? And basically there are huge gaps in our knowledge here. We've pretty much got no idea how these 3 states, 3 levels are tied together. There's a bit of an idea, this is taken from Parrish's thesis, I think, about how the first 2 levels connect. It's quite a complicated diagram, and you don't need to understand it, but what's going on here is these are the 5-HT neurons from the RAF nucleus. This is the serotonin being released. This is the 2A receptor here, and these are these secondary messengers, so AA and 2AG being produced at the receptor. And the idea is that psychedelics affect these waves of 2AG and AA, making them out of phase or changing their phase, and therefore changing how they interact with this glutamate neuron here, changing how that neuron fires, and then changing how this neuron fires. And this neuron is a layer 5 pyramidal cell. So that, again, is extremely speculative. We don't know if this is the case or not, but at least it's an idea about how you could get from the receptor level to the kind of local neuronal level. And finally, just to some questions that really remain outstanding. What role do the 2C and 1A receptors play? Like, we don't really know a lot about this. We don't know whether 2A is sufficient for psychedelic effect or only necessary. It might be the case that you need 2A, but you also need 2C as well. And we don't have selective psychedelic agents that are purely 2A. There are a couple that are in development, some rigid analogues of the NBOME series, which have very, very high selectivity for 2A over 2C. So that will be helpful for exploration of this. And again, we don't really know what role 1A receptors play. They obviously are heavily involved with the tryptamines, but we don't know how that changes the experience. And there's only been that Strassman experiment. Can we trust animal models? Rats are not little people in furry suits. I think this is a really favourite quote of mine. I can't remember who said it originally, but all these experiments tend to be drug discrimination, where you're training a rat to press a lever when it's given LSD, and you train it and train it until it knows to press a lever to get a food reward when it's given LSD. Then you give it another drug, and you're basically saying, how much is this drug like LSD? Have I given you LSD? And the rat presses the lever if it thinks it's LSD, and it doesn't press the lever if it thinks it's not LSD. But the question here is, like, what's actually going on in the rat's brain? What effects is the rat experiencing? And it could be that what the rat experiences as 2A agonism is actually quite different from what we regard as psychedelic. And they don't care about the 2C activation. They just go, I'm getting 2A, and that's like a kind of, it's like a stimulant buzz or something. I'm just pressing this button because it feels like 2AE. But in humans, maybe we need 2C as well, and that's missing part of the psychedelic experience. So it's important to, when you look at these animals, try and work out how they translate, and basically human experiments are only the way forward. And finally, why is LSD so potent? All this stuff I've shown so far just doesn't show why LSD is so potent. It's a huge question mark, and it's probably one of two things. Either it's to do with the other receptors that it's activating, the dopamine or histamine or any of the... LSD is a very, very promiscuous molecule. It activates a lot of different receptors. So it might be something to do with those extra receptors, or alternatively, it could be because it's activating another secondary messenger pathway in 5-HT2A that we just haven't discovered yet, and that might be the cause of LSD's potency. And I think that's me. So